So in this meeting, I presented results from a single-cell RNA sequencing study we conducted on tumor and immune cells from 30 patients with asymptomatic Waldenström's and overt Waldenström's. Because we had the ability to look at both tumor and immune cells, that allowed us to answer both questions about tumor intrinsic features as well as tumor extrinsic immune features. So on the tumor intrinsic part, we again observed that there is unexplained heterogeneity within MYD88 mutant tumors, more than we just understand right now, more than we can just explain by CXCR4 mutation status or recurrent copy number variants. And indeed, when we look at transcriptional features, we again identify signatures similar to the memory B cell and plasma cell-like signatures other people have been describing. With two key signatures, one marked by CXCR4, and that is a signature that is associated with a higher risk of progression, although not entirely explained by CXCR4 mutations. And I think that's maybe a next step we need to take here to really understand what is the intersection between high levels of CXCR4 expression versus CXCR4 mutations. And I think a question that came up and is very interesting is not all CXCR4 mutations are made the same, and therefore, what CXCR4 mutations lead to what downstream effects would be also very important to study. And then a signature marked by DUSP22 and CD9 that is probably most similar to the plasma cell-like signature that other people have been describing, that we find is again mostly mutually exclusive with CXCR4 mutations, therefore more likely to respond, for example, to ibrutinib likely, but also associated with the presence of lymphadenopathy. And this is important because CD9, which marks the signature, is a gene that lymphoma cells like to use to interact with follicular dendritic cells within the lymph node. And therefore, it starts making sense that this is a phenotype that likes to stay within the lymph node, and therefore the patients present with lymphadenopathy, which is a criterion of progression. And then within the immune system, we see significant alterations in bone marrow immune cell composition, most of which actually, or a lot of which, seem to be specific to Waldenström's. When we compare them, for example, to smoldering multiple myeloma, which is another type of cancer that arises in the bone marrow, there are similarities, but there are also, distinguishing abnormalities between the two. So we feel this represents a paradigm shift when it comes to thinking about the usefulness of immune profiling for diagnostics, because this far in cancer, we either will look for the cancer cells themselves or we look for products of cancer cells. But in this case, you can just look at the immune microenvironment and determine based on that signature whether the individual is a patient and what kind of cancer the patient may have. There are two key immune hallmarks we identify in patients with Waldenström's. The depletion of activated T and NK cells and the depletion of interferon-stimulated T and NK cells. And we find that this is, again, a specific feature of Waldenström's. It is not seen in smoldering myeloma. And based on our data, we hypothesize that this is a reflection of a hypo-responsiveness to interferon rather than due to the lack of interferon, for example, in the blood of these patients. And that if you take the T and NK cells outside the body of the patient, put them in a dish and stimulate them with interferon, you can actually partly reverse that defect. It never quite catches up to what we see in healthy donors, but you can partly reverse it, and we have data showing that this does impact immune cell functionality. So we can think of this as maybe setting up the stage for an immune dysregulation targeted treatment in patients with Waldenström's.